Module 1, Part 5. In Part 5 of Module 1, we are going to talk about electron microscopy. So far, the discussions have been light microscopy and variations on light microscopy. Our beam of radiation was light. Now I want to switch to electron microscopy, where the beam of radiation is electrons and where the wavelength of a beam of electrons is substantially smaller than the wavelength of light, producing a spectacular resolution with the electron microscope. So let's calculate the resolution of a typical electron microscope using the formula. So what is the best resolution one can obtain with a transmission electron microscope, or TEM? As you can remember from the light microscopy, the best resolution was about 0.2 microns. The wavelength of electrons is 0.005 nanometers. The ensign alpha of the best electrons is 0.0305. So using these numbers in the formula for resolution, you can get a resolution of transmission electron microscope of 0.1 nanometer, or 2000 times better than a light microscope. So this figure gives you an overall very clear idea of the imaging techniques available for the biological samples from, say, a cell to the atomic resolution. The horizontal, the x-axis, is the spatial resolution. It starts from the left at 0.1 angstrom, or 0.01 nanometer resolution, which is the atomic resolution, where you can visualize a single atom, to the cellular and tissue imaging resolution to the far right. The y-axis is the temporal resolution, which determines how fast of a dynamic event can be captured by an imaging technique, starting from the top at the femtosecond range to minutes range at the bottom of the scale. So let's take a look at the spatial resolution of electron microscope. The focused ion beam of the ACM, or scanning electron microscope, is in the range of 2 to 10 nanometer. This range is great for visualizing relatively larger surface structures. The dynamic TEM, or transmission electron microscopy, can visualize at the twofold higher resolution than ACM to up to about 1 nanometer range, and can visualize ultra-thin section samples as well. But if you see the cryo-TEM in the dark blue with the mind-boggling computational power, you can even visualize a single atom at the resolution of 0.1 angstrom, or 0.01 nanometer. This is how we visualize the biological macromolecular detail of the proteins, carbohydrates, nucleic acid, atom by atom. So take a look at the comparison of the setup of the microscope. The transmission electron microscope is very similar to the light microscope, except the beam of radiation is made up of the electrons. So let's review the light microscope. There is a light source, which may be a laser in the case of a fluorescence microscope. This light is focused on the sample using a condenser lens and collected and magnified through the objective lens, and the image is viewed through an eyepiece. The setup of a transmission light microscope, the source of the electron is a heated element. There is a condenser that focuses the electrons onto the specimen, an objective lens that collect them, and a projection lens which put the image on the screen. Here is another rendition of the working schematics of the TEM on the left and a typical TEM setup on the right, and showing the placement of some of the key parts of the instrument. So the detector is a phosphor screen, and it lights up if an electron hits it, just like an old box TV. You can directly visualize the phosphor screen with a binocular eyepiece, as shown in the figure. There is also a camera under the phosphor screen, which captures the image and projects into the monitor as well. So we talked about the sample preparation and staining in light microscopy earlier, and how that improves light microscopy. Sample preparation is much more complicated for transmission electron microscopy. That is the main disadvantage of TEM. The complicated sample preparation. The images are great, but it takes a long time to prepare the specimen. You use the same 3 steps: fixation, embedding, and sectioning and staining. The fixation is carried out using the cross-linking agents such as light microscopy, such as glutaraldehyde, but there is a second fixation compound that is often used for TEM, and it is osmium tetroxide. It is very important because it binds and stabilizes the lipid bilayer, which is very important in TEM because when you section, you use much thinner layers than light microscopy. But the osmium tetroxide is also electron dense and it serves as a stain, which I will discuss shortly. The second step is embedding, and the specimen must be sectioned much thinner than the light microscopy because the penetrating power of electrons is much lower than light. So the layer has to be much thinner for the electrons to get through the specimen and be detected on the computer screen. Embedding is done by dehydration of the sample, and the sample is encased in a solid plastic block. Sectioning is done by a sophisticated microtome known as an ultramicrotome or cryomicrotome that gives section of 50 to 100 nanometers in thickness. After sectioning, the samples are stained with heavy metal stains such as uranyl acetate, lead acetate, phosphotungstic acid, and osmium tetroxide, which can also be used as fixative as mentioned earlier. The reason for using heavy metal stains is because they are electron dense, means the electrons will not be able to pass through them and the stains will appear dark. So the purpose of staining is each stain has a different affinity for the subcellular structure and will bind to different structure and organelles at varying degrees. This will give the electron micrograph the black and white contrast of varying degree. The area with lots of stain bound will be very dark or black, moderately bound area will appear gray, and the area without any bound stain will appear white. We will visually see this in the next slide. Note that all electron microscopic stains are called negative stain as they are negative or dark as the electrons can't pass through them, in contrast to in a light or positive background through which the electrons pass without obstruction and hit the detector. Different organelles in the cell will pick up the stain more or less efficiently. The organelles that picks up the stain poorly will allow electrons to pass through them, and those electrons will reach the detector and appear very light. An organelle that absorbs the stain very efficiently will deflect most of the electrons. All negative stains contain heavy metals and are very dense, which means it deflects the electrons and very few electrons will get through to the detector, and that portion of the specimen will look very dark compared to this. Here is a typical transmission EM image. These are cells from an onion root. You can see spectacular detail. You can see the nucleus, you can see the mitochondria, amazing detail. So how do you get the contrast? For example, the dark nucleolus in the nucleus versus the vesicles that appear white. So the contrast depends on 2 things. One, the type of macromolecules found in the various areas and organelles in the cell which bind to the negative stain differentially. The second is the greater the atomic number of macromolecules in that region of the cell, the greater the electron scattering, which produces greater contrast. Those regions will be darker at a varying scale. Here are some of the more representation of the TEM images. On the left, you can see the coronavirus with their spike proteins sticking out, and that's why they are named, so having a corona. As you can see, the background is stained relatively dark with the negative stain, and thus the virus is lighter in contrast, but there are some varying degree of electron-dense material on the virus and gives some degree of variable gray shades. On the right is the rough endoplasmic reticulum of a cell and some other organelles. And you can see the membrane of the ER is very darkly stained, while the lumen of the ER is less electron dense, thus appearing lighter in contrast. Here is also an amazing electron micrograph showing a neutrophil is engulfing two bacteria by phagocytosis. This happens in our body every day, all the time. Remember, there were variations on fluorescence microscopy known as immunofluorescence microscopy, which relies on an antibody that binds to your protein of interest. There is a similar variation known as immunoelectron microscopy. It relies on an antibody that binds specifically to your protein of interest, and a secondary antibody is not connected to a fluorophore but covalently connected to a gold particle. Because gold is very electron dense, it can deflect the electrons and make those regions of the cell very dark, so you can identify specific proteins on the transmission EM. There is a variation. If you don't have a gold-labeled secondary antibody that will bind to the primary antibody, you can obtain commercially available gold particles that are bound to a protein known as protein A. The protein is isolated from Staphylococcus aureus bacterium, and protein A specifically bind to the Fc portion of antibodies. This is the second way is to label your primary antibody for visualization on the electron microscope. In this example, this is an antibody that specifically binds to catalase, which is found in the organelle known as peroxisome. So how does the gold particle work? As mentioned, gold is very electron dense, so if the peroxisome is stained with the anti-catalase antibody with the gold labeled protein A, then the gold particle that are bound to the organelle will deflect the electrons and will identify that organelle. So here is an example. These are the peroxisomes, and you can see these tiny black dots, which are individual gold particles that are identifying the catalase found in that organelle. Cryo-electron microscopy is a state-of-art technology, multi-million-dollar instrumentation. It is very powerful transmission electron microscope, and what we mean by that is the electron beam producing coils generate electron beams with the voltage application of up to 300 kilovolt or 300,000 volt, which is almost 3 times higher in voltage than ordinary TEMs. The higher voltage means the stream of electrons have more energy and penetrating power. In that way, it is a very powerful TEM. The sample preparation and imaging technique is different than the conventional TEM sample preparation that we have seen earlier. In cryo-EM, the sample is frozen very quickly in water with liquid nitrogen at minus 196 degrees Celsius. This extremely rapid cooling forms the amorphous or vitreous ice, which is non-crystalline, and hence the name cryo-EM. The sample is kept at the cryogenic temperature throughout the image data collection. So what is vitreous ice? When water forms ice by normal cooling, it forms crystal with crystal-like lattice, and any living sample with water will be destroyed by these sharp crystal edges. But a rapid cryocooling method does not allow the crystal lattice of ice to form, and it forms a glass-like amorphous ice or vitreous ice, which preserves the specimen entirely in its original form. The sample does not need any fixation and staining. The natural electron density of the sample is sufficient for a cryo-EM to collect meaningful data. But there is another step, that is the difference in image data collection. Ordinary TEM, a single plane of image is captured in one single orientation. In cryo-EM, the sample stage is rotated up to an angle of plus 70 degrees to minus 70 degrees with 1 or 2 degree of increments. So each time, each frame of image have a slightly different orientation of the sample. This slight difference in each individual image is processed and analyzed with highly sophisticated mathematical algorithms to produce stunning three-dimensional images even at atomic resolution. Earlier conventional TEM could be used to reconstruct 3D images, say of a cell, but it is extremely labor intensive. To do that, you have to serially section a cell embedded in a block, arrange each slide of the cell in order to precisely say top to bottom, and then stain each of the section individually and take a static TEM micrograph of each of them, and then reconstruct a 3D image computationally, merging each individual image. All this labor-intensive months of work is solved with way more higher resolution with this state-of-art cryo-EM. You are familiar with the term CT scan or CAT scan in the medical imaging. It stands for the computer tomography or computer-assisted tomography, which gives a 3D image of the tissue in a patient's body by scanning tissues in different planes from which a 3D image is formed. The same way, the cryo-EM images are collected by varying the stage angle a little as mentioned earlier, and from that, a 3D image or tomographic image is formed as shown above. The top picture shows the yellow nuclear envelope with the purple nuclear pore complexes. In the bottom left and right, one individual pore complex is visualized in the bottom and the top orientation. Please note that these images are actually pretty low resolution. As I mentioned before, the cryo-EM techniques is capable of visualizing at the atomic resolution as well. So transmission EM requires extremely thin sectioning and provides excellent 2D information and detail, but loses 3D information from the specimen. We are not talking about cryo-EM though, just regular TEM. Remember, confocal microscopy solved that problem with fluorescence microscopy. The solution for that problem for transmission electron microscopy is a different kind of electron microscopy known as scanning electron microscopy. It does not require the sectioning of your specimen and will provide a 3D image and information of the three-dimensional characteristic of the sample. ACM, you can use whole cells or unsectioned tissues which are fixed, dried, dehydrated, and coated with a heavy metal such as a platinum, which will deflect the electrons. Please note that the transmission EM negative stains were salts, acids, or oxides of the heavy metal, but they were not pure heavy metal. In ACM, to coat the sample, a block of pure heavy metal like platinum or gold is evaporated to form a fine layer of coating of nanometer thickness over the sample. Here is the setup of a scanning EM and compared to the light and the transmission EM. There is a heated filament tungsten. This is the source of electron which is focused on your sample, which is not sectioned and coated with a heavy metal. The heavy metal coating will deflect the electrons, will be picked up by a detector and transferred to a viewing screen on a computer. The degree to which the certain portions of the sample deflect the electrons will give the image the 3D dimension. Here is another rendition of the working schematic of the ACM on the left and a typical ACM setup on the right. and showing the placement of some of the key parts in the instrument. Note that there is no optical visual port or eyepiece like you have seen in the TEM in an ACM setup. The deflected electrons are detected by the detector, and the image is processed computationally to project only on a monitor or a camera. Here is a comparison of scanning EM with a transmission EM and a Nomarski interference microscopy, which uses polarized light to give a pseudo-relief image of what you are looking at. So these are cilia projections from hair cells in the inner ear of a frog. The upper right image is the Nomarski or DIC. You get some idea of the three-dimensional structure of this projection from the hair cells. Remember, the resolution is approximately 0.2 microns. The transmission EM on the panel show the amazing detail, but no idea about the three-dimensional nature of the structure. The left image is the same structure on ACM, so you sacrifice some detail in the ACM, but can obtain some excellent information about the 3D nature of the structure and the materials. Here is another ACM image of the lower respiratory tract. This concludes part 5 of the module 1.